Morning friends, my name is Sudhi Bitaria from University and today's my topic is enzymes. Right. Let's see, in today's session we will discuss about the what is enzymes, chemical nature of enzyme, enzyme power, active site of enzyme and effect of different parameter on enzymes. First of all, temperature, pH, enzyme concentration, substrate concentration, cofactor and inhibitor. Let's see what is the enzymes. Enzymes are a biocatalyst and enzymes are a complex chemicals. Enzymes, that is the protein composition of enzymes, that is the protein. Complex chemicals, that is control the reactions in the living cells. Right. In the living cells, without enzymes, metabolic activity is not possible. It is a biochemical catalyst. Catalyst, the word catalyst, that means which increase the speed of the reaction, which maintain the speed of the rate of the reactions. Right. The chemical in which the enzymes work is known as the substrate, on which the enzymes, for example, hexokinase, right, hexokinase which react onto the glucose, right. So hexokinase, that is the enzymes, and glucose, that is the substrate, and enzymes that combines with the substrate to form a short-lived enzymes, or we can say substrate complex, and once a reaction occurred, the complex breaks down into the product. Right. Here you can see the E, that is the enzymes, plus S, substrate, gives the E as ES, that means the enzyme substrate complex, and it's converted into the enzymes P, E and P, that P indicates the product. The enzymes remains unchanged during the reactions, right, and this enzymes which is again interact with the another substrate and it forms the product. Here you can see the diagram. This is the enzymes, right, this is the substrate and there is enzyme substrate complex, right, and then enzyme substrate complex converted into the enzymes and product, and this enzymes which is released, and this is the unchanged and again reacted with the another substrate. Right. The role of the enzymes in the living organisms, and many of the complex chemicals in the living organisms need cannot be made in a single reactions. Instead of a species of a complex reaction occur one after the another, right, forming a metabolic pathway. Here you can see the A, B, C, D, right. Enzymes control the cell metabolisms by regulating the how and when a reaction occur, right. This is a complex reaction. The final product is D, right, the final product is the substrate D, right. A, with the A, enzyme 1 is reacted and converted into the product B. With the product B, enzyme 2 which is reacted and converted into the product C, and this product C, that is the substrate for the enzyme 4, E4, right, and it is converted into the D. Next one is the chemical nature of the enzymes. Enzymes is a protein, and protein there is many class, there is primary, secondary, tertiary and quaternary protein. So enzyme is a globular protein. They have a complex tertiary and quaternary structures in which the polypeptide chain folded around each other to form a roughly spherical or globular shape, we can say a ball-like shape, right. Enzymes have a several important property. Enzymes are specific, that means each enzymes have a catalysis on one reactions, right, that means which is specific for the substrate. Enzymes combines with the substrate to form a temporary enzyme substrate complex. Enzymes are not altered or used by the reactions, they are catalyzed or so used again and again, right. Enzymes are sensitive to temperature and pH. Many enzymes need cofactor in order to function. Enzyme functions may be slowed down or stopped by the inhibitor. What is the enzyme's power? All the reactants need to have a certain energy before they will react, right, and this is like an energy barrier that it has to overcome before a reaction will occur. It is known as the activation energy. You can see here the this is the without enzyme and this is the with the enzyme, right. With the enzymes the activation energy required is a lower, and with the enzymes without the enzymes activation energy required it's high, right. Enzymes, that is the organic catalyst, and all organic catalysts to lower the energy barrier, that means which decrease the activation energy, and these enzymes do not participate in the reactions, right. This is the diagram of the enzymes. Amount of the energy which is utilized against the reaction of the enzyme. This is the without the enzymes, right, activation energy that is utilized, that is higher, and this is with the energy, activation energy, that is low. Enzymes active site. Enzymes active site. Enzymes have a specific region at which the substrate bind, right, and this site known as the active site. This is the active site of the enzymes, right. Enzymes are substrate specific, and specificity varies enzymes to enzymes. When a substrate bind to the enzymes, that means ki at the active sites they form the enzyme substrate complex. Changing the active site, the changes into the shape of the active site will result in the loss of the function. That means ki denaturation of the enzymes, and it is also happen when the higher temperature or higher pH we will provide. You can see denaturation of the enzymes occur, and enzymes are the protein, right. That means ki denaturation of the protein occur, and they lose the functions, right. Next one is the effects of the parameter on the enzyme action. The first parameter, that is the temperature. Temperature speed up all the reaction, but the site of the denaturation of the enzymes also increases at the higher temperature. You can see here the percentage maximum activity against the temperature. When the temperature will increase, the enzyme activity will increase. And at the some temperature, above the optimum temperature, if we increase the temperature, higher temperature breaks down the disulfide bond that holding the tertiary structure of the protein that is present into the enzymes. This destroys the active site and therefore make the enzymes non-functional. This is the lower portion, too cold for the enzymes to work, too hot for the enzyme to work, and this is the optimum temperature for the enzymes. Here there is a four diagram, four shape, first one, second one, and third one, right. This curve in the blue shape, blue color, isolated from an organism, so that is the organisms that is live into the habitant of the cold area, right, that is the psychrophiles. Next one is the curve in the red shape, plays an enzymes isolated from the digestive tract of the human, that means at the 37 degrees Celsius, and the curve in the green, that means that grow in the area there is where there is a very high temperature, that is thermophile. pH of the enzymes, like all the proteins which is affected by the pH, right. If pH, that means measurement of the hydrogen concentration. If the higher pH value, that means higher pH value, that means ki higher pH value, that means 0 to 7 acidic and 7 to 14, that is basic, right, as which measure the acidity and alkalinity, right. And the green color, the green curve, that is for the pepsin that digests the protein in the stomach, right. And the red curve that show the activity of the arginase that breaks down the arginine to ornithine and used into the liver. The next one is the enzyme concentration on the enzyme action. Assuming that the enzyme amount of the substrate is not limiting, and an increase in the enzyme's concentration causes the increase in the rate. If the enzyme concentration is high and we will provide the substrate, then the activity of the enzymes will be increased. Here you can see the graph. Next one is the substrate concentration. Assuming that the amount of the enzyme is constant, right, an increase in the substrate concentration causes the diminishing the increase in the reaction rate, right. Here you can see the substrate concentration. When the substrate concentration is higher, right, at this stage the substrate concentration is high, then the V maximum will be high, and this is the substrate concentration is low, then the V maximum is lower. Next one is the effect of the cofactor on the enzyme action. Sometimes enzymes need the cofactor to increase the activity, to increase the catalytic activity of the enzymes, and this cofactor may alter the shape of the enzymes slightly to make the active site functional or to complete the reactive site, right. Enzyme cofactor include the coenzymes, organic molecules, and the activating ion Na plus and K plus. Vitamins are often act as a cofactor, vitamin B1, vitamin B6. Next one is the enzymes, nature of the enzyme inhibitor. Enzyme inhibitor may or may not reversibly. There is two type of the inhibitor, reversible inhibitor and irreversible inhibitor. Inhibitor is temporarily, reversible inhibitor, that is the temporarily bound to the enzymes, that is bound that bind to the enzyme at the active site, right, that is used as a mechanism to control the enzyme activity. And the irreversible inhibitor, that inhibit may bind permanently and which causing the permanently deactivator. And in the reversible inhibitor there is a two type, competitive inhibitor and non-competitive inhibitor. There is a work in two ways. Competitive inhibitor that competes with the substrate for the active site and blocking it or preventing the attachment for the substrate. And in the non-competitive inhibitor, the inhibitor bind to the enzymes not at the active site, but the that bind to the enzyme with another side of the active site, but it alter the shape of the active site, so the enzymes cannot bound to the active site, right. This is the allosteric inhibition. Thank you.